Most people think about Parkinson's disease through sort of a genetic or molecular lens. So for me, it's alpha-synuclein, it's LOCK2, mitochondrial, autophagy. What made you look beyond genetics and focus specifically on the microbiome in Parkinson's disease?
Yeah, thank you so much for having me, Aaron. That's a great question. So there's been a long history actually of the gut microbiome and the brain axis. So that there's a very close relationship between processes that are happening in the gut and what actually happens in the brain. So there's been increasing evidence over the last couple of years, particularly in that context around the microbiome, but more generally just in terms of the gut-brain axis, that a lot of things that are happening in the gut has an impact on the brain. So a lot of signaling that communicates between the gut and the brain. And because of that, a lot of the changes that happen in the gut, which we can get into the microbiome, but the microbiome plays a big role there. A lot of the signaling molecules and things that these microbes produce affect processes in the brain as well. So that's been a whole branch of research in the last couple of years, and the microbiome plays a central role in that. And then there's obviously in clinical conditions in neurodegenerative diseases, there's a lot of evidence around how the gut might impact these diseases, which includes Parkinson's disease, but also other neurodegenerative diseases that the gut plays a major role in these diseases actually.
So let's focus on, let's say, the clinical phenotype and the symptoms, because most people associate Parkinson's disease with motor symptoms. But interestingly, your work focuses on cognition. And so why was cognitive impairment the right phenotype to study in relationship to the microbiome specifically?
Yeah, so as you mentioned, especially in terms of Parkinson's, most people associate this with motor symptoms like tremors and these kind of symptoms. But a lot of these patients also do suffer from cognitive impairment. So especially in later years, after about I think 10 to 20 years, most of these patients do develop dementia. So it is a major non-motor symptom in patients with Parkinson's disease. And because it's quite difficult still to diagnose cognitive impairment, that was one of the reasons why we focused on cognitive impairment, because it's quite difficult to diagnose clinically. There's sometimes quite often there's not very robust biomarkers to be able to do this. So it's very much sometimes up to clinical clinicians' interpretations and these kind of things. That's not just in Parkinson's, but in general, cognitive impairment and cognition is quite difficult to diagnose. So in that context, we were looking for some sort of biomarkers between different stages of cognitive impairment and not just focus on motor symptoms. Although we take into account some motor symptoms as well, we specifically wanted to see if the microbiome, because there was a lot of evidence before our study between Parkinson's patients and healthy patients. So we know that the microbiome plays a role there, but not necessarily in different stages of cognitive impairment, which was not that known beforehand.
I got 2 points to that. So clinically speaking, I'm not sure if you can answer that, but let's try it. With regards to the relationship to motor symptoms and say cognitive impairment, is it motor impairments are first sort of the early clinical signs and then afterwards you've got cognitive impairment? Or do you think maybe they may start sort of side by side at the same time, and just maybe due to the subjective nature in the clinic when you're looking at cognition versus motor impairment, it's just more difficult to detect cognitive impairment, and that's why it's detected later in comparison?
Yeah, that's part of the issue that this is a sort of a spectrum of cognitive impairment, and especially early on, this is quite difficult to diagnose, and whereas there's quite good diagnosis for motor symptom impairment, whereas with cognition this is sometimes quite a bit more difficult to diagnose. So again, I'm not a clinician, but these symptoms do appear sometimes across a spectrum, which is what we studied as well, and they do appear potentially a bit later on in the disease progression. And it's also quite something that we can touch on later as well. But what we also tried to do in the study is, because it is quite difficult to disentangle these two things as well, right? So to really know what is because of the cognition impairment, potentially what is because of the motor symptoms of the disease itself, right? Because the motor symptoms is one of the primary sort of symptoms. We is it really possible to disentangle? That is quite possible because they do go hand in hand, right? It's not a very clean line of the one and the other, which one appears first and which one not. But in general, these patients have sort of early mild cognitive impairment, and later, like I mentioned earlier, most of these patients within about 10 to 20 years do develop full dementia. But this is most often a bit later on in the disease. Yeah.
Sort of moving forward to that line of thought, maybe mechanistically, I'm just hypothesizing here, dependent on what areas of the brain are first predominantly affected by neurodegeneration, which then correlates to, let's say, the clinical phenotype. And so, say, for instance, if the olfactory system or the olfactory epithelium is affected, then maybe the first clinical sign would be lack of smell, for instance. And so maybe that differs from different types of Parkinson's and different types of severity. But yeah, I guess mechanistically, it's sort of difficult to disentangle because Parkinson's disease, especially sort of sporadic, it's so sort of heterogeneous in not only its mechanism but also its clinical phenotype, I think, anyway. But I'm just spitting sort of thought experimental ideas out there. But so before we sort of delve into your paper, let's talk definitions. So what is the microbiome? What does it actually mean, and how is it different to just sort of talking about bacteria that would use for doing a protein purification prep or something? Like how's it different?
Yeah, so the microbiome per definition, or a microbiome, you can start with a microbiome, and then we can get to what we sometimes refer to as the microbiome, which we actually then refer to the human microbiome. But a microbiome, per definition in general, is a collection of microorganisms that function within a community, right? So they either cooperate with each other to live sort of together in an environment. So that's a microbiome in general, which can be in different environments. So this can be, in our case, when we look at the microbiome, but it can also be in the environment or in animals or plants or whatever. There's collections of microorganisms that impact the host. In the context of the microbiome, the human microbiome, so a lot of people would obviously have seen all the stuff in the news in the last couple of years around the gut microbiome. So that is the collection of microorganisms that live within our gut. So within the gut, they have really important functions in terms of metabolism, but also immunity. So that's the collection, sort of, they together as a collection aid in these functions within the host, right? So with more to your point, so the difference between that and just talking about bacteria is, first of all, the microbiome is a very diverse collection of bacteria, but it also includes other microorganisms. So which we didn't look necessarily in our study, but it also includes fungi and also includes viruses and some other sort of micro eukaryotes even. So it's all microorganisms together. It's not necessarily just bacteria. So that's the gut microbiome. But in the course of the last couple of years, we've also, in the context of the human microbiome, there's been a lot of work around other sites, and that is also really important. So a lot of work in which we also did, obviously, in our work around the oral microbiome. So that also plays a major role, even when you think about digestion in the context of the gut. So your mouth is sort of the first point of entry of diet intake. So a lot of the microorganisms, they play a role, but they also play a role in different diseases. And the oral microbiome is very specific niches in terms of the teeth versus the tongue versus the saliva. There's different composition, different types of bacteria that live within these different environments. And then there's also a lot of work around the skin microbiome as well, and that also plays a major role in different sort of skin disorders. And then also, so there's a lot of work around the microbiome in general, but per definition, it's basically the collection of microorganisms in the gut, which we refer to as the microbiome, which live in the gut, reside in the gut, and help with a lot of different functions within the human physiology.
You mentioned the skin microbiome, and this has popped up. So we're going to talk about later about metagenomics and proteomics and all those. Have you heard of Sebenomics? I think it's called like sweatomics.
No, I haven't done that.
You got to search this up. There's I remember I went to a toxicology conference a couple of years ago, back when I was at Cambridge for my PhD, and we heard about this woman who could smell Parkinson's.
But yeah, that I've heard of, yeah, but
Yeah,
not.
that's just based on Seebemomics, where like
Okay.
yeah. So it's really cool, like how just sequencing different, let's say, bodily fluids and
Yeah.
whatnot can really provide insight.
Yeah, I didn't know I wasn't familiar with the term, but I mean,
Mm,
there's a lot of work around those things, around different smells and stuff, which the microorganisms are actually responsible for that.
mm,
So that
mm,
was, as far as I remember, the hypothesis about the smelling of the Parkinson's was that these bacteria secrete very specific compounds, and that
mm.
is what sort of creates that smell. Yeah. So this
Yeah.
is quite interesting. It's quite well. There's a lot of great work around that as well. Yeah.
It's, yeah, I thought that was really cool. But anyway, let's talk about your study and your work, because I thought it was really cool. And so, what was the primary goal of the study when you decided it? And how did you also structure the cohorts of different types of patients you use? Like, how did you design the study as well?
Yeah, so I can first mention actually that so there was a really nice collaborative study actually with some of our colleagues in Turkey. So they collected the samples actually there in Turkey. So it was a Turkish population. So the primary aim for both from the sort of more computational side, but also from the clinical side was really, as we already sort of touched on, was about this difference in cognitive impairment across Parkinson's patients. So the primary aim was to see whether or not we can identify different microbiome signatures across that scale. So across patients with mild cognitive impairment and patients with dementia. So that was the primary aim, to see if the microbiome actually changes between that. Because before our work, there has been a lot of work on showing how the microbiome changes in Parkinson's. So, and that was also shown before across different scales of motor symptoms, for example. So we knew that there's a lot of changes in the microbiome in different species that are there in Parkinson's, but not necessarily across cognitive impairment scale. So that was the main aim, is to see, when you go sort of from healthy controls to mild cognitive impairment to dementia, does that microbiome composition, to the species that are there, actually change across the scale? That was the main aim, and that's how the study was set up as well, right? So we collected samples from patients that with mild cognitive impairment. So they were clinically assessed on different scale, on different tests and things for cognitive impairment, and they were classified by a clinician as mild cognitive, that they have mild cognitive impairment, and then another set of patients that had dementia. So when you look at the different scores that are used, clinical scores, again, I'm not that familiar how these scores are set together, but when you look at these different scores, they have different cognitive ability, going from mild cognitive impairment basically to dementia. So that was how the study was set up. And then and then there was also a healthy control arm, which were also collected during the same studies. They From these patients, we collected stool samples to assess the gut microbiome, and then also collected saliva samples from these patients to assess the oral microbiome, which we can get into in a bit as well, why we did that. But so that's basically the data that we've worked with. So then be able to assess in these different groups what species are basically there, and to which level they are there. That was the basic setup. Yeah.
So just to clarify this as a little quiz, there are three patient groups, well, three groups. So healthy control, mild cognitive impairment, Parkinson's disease, and then Parkinson's disease dementia patients, right? And you received, or you took out the samples of the oral microbiome and also the gut microbiome, and that was through like a saliva test, like taking saliva for the oral microbiome,
Yes,
and
saliva.
then fecal
Fecal.
samples or stool samples for the gut. Why did you look at both the gut and the oral microbiome?
Yeah, that's also a great question. So, like I mentioned earlier, so in the sort of microbiome field, there's a lot of work done on the gut microbiome, but the oral microbiome in general is we've seen more and more that the oral microbiome is also plays a very important role in different diseases. So we found in some of our other studies, for example, this is not in the context of Parkinson's, but we found, for example, in a lot of chronic diseases that there's something that we call sort of oral gut tropism, that a lot of the oral species in these that are more oral resident species that we know they are mostly reside in the oral microbiome, that during certain diseases we've done some work on, for example, liver disease, that a lot of these species translocate sort of through the gastrointestinal tract, and then we pick them up in the gut. And then this is quite good signature in a lot of these diseases. So you kind of see, for example, in liver disease with progression in cirrhosis, then all of a sudden some of these species appear within the gut. And the hypothesis is that they kind of change their niche as well. So they kind of, the functions that they perform going from the oral microbiome, where there's, for example, a bit more oxygen and things like that, going to the gut where it's a anaerobic environment, and then the pH changes, everything. So they niche kind of change, and then they potentially become pathogenic. So then, whereas in the oral microbiome they might not be, and then they translocate to the gut, and then they might become. There's a lot of work still to be done on that in terms of functions. That's sort of the hypothesis. So that was the one side of it, whether or not this sort of oral gut tropism also happens in Parkinson's. And then apart from that, there has been before us as well a lot of work on other neurodegenerative diseases as well, and the oral microbiome. So there's been, you might be familiar in Alzheimer's around work around Porphyromonas gingivalis, where some of the species in the oral microbiome are sort of, they found that they translocated to the brain actually, and they do certain things there. So there has been some evidence around not necessarily Parkinson's, but in things like Alzheimer's, that very specific species like Porphyromonas gingivalis do play a role and to contributing towards disease progression. And so that's why we also wanted to look at the oral microbiome in general, because it is much less studied in Parkinson's. So that was the hypothesis. So whether or not potentially this tropism happens, or if that doesn't even happen, whether or not the oral microbiome also in these cognitive stages do also change, and if there are potentially some markers that we can look at that are because of the cognitive impairment, just merely because there might be oral microbiome changes.
So 2 quick questions before we move on to the main takeaway of the paper. How many sort of different species or populations of different types of microorganisms do you think inhabit the oral microbiome and the gut microbiome? And secondly, you mentioned this sort of translocation of microbes between the gut and the oral microbiome. How do you know how that, because like, how does one species or one microbe sort of move between, because they're very far apart, and like, is it via the bloodstream? Like, what's going on there?
Yeah. So in terms of numbers of species, one of the issues in really determining how many different species we are talking about goes also back a bit towards the technology, right, which we haven't talked about yet, but around metagenomics. So how we actually capture these microbes, because a large proportion of these microbes, especially in the gut, is not culturable, right? So we can't culture them in the lab, whereas in other cases you can. So certain of these species you can't culture. Some of them is just a lot more difficult to culture within a lab, and with culturing, you know better than I do, but I mean, is literally basically growing these microorganisms on a plate, right? So because the conditions might differ, they're quite difficult to grow. Some of them only grow in biofilm, some of them don't, some of them you don't grow at all, struggle with anaerobic versus aerobic conditions, all these kind of things. So with metagenomics, which is the technology that we use, we capture all the DNA within a certain community. So from the fecal material, we take out all the DNA that includes the microbial DNA, and then we sequence the microbial DNA. So then we basically look at the DNA that is there of all the microbes, and then through a lot of computational efforts, we then basically reconstruct the genomes of these organisms, right? But we are still never 100% certain if we construct full genomes. We capture parts of the genomes. So it's a computational process again, because if we can't culture them. So for the microbes that you can culture, you can culture them, take out the DNA, and then sequence that, so you know what's the full genome. But in this case, we reconstruct them computationally, these genomes. So that is the one difficulty in knowing exactly how many species we're talking about. But in the gut, with what there's still a lot of a part of it that we haven't been able to do. But that we still don't capture necessarily. But at least from these studies, we are talking about sort of the number of species, not necessarily how many in total they are, but the different types of species in essence might be, I mean, in the orders of hundreds, right? So we're talking about 2 to 500, whatever different species that's not the numbers, right? So they're obviously there in much bigger numbers, but that's sort of the different number. And again, this changes across that changes across populations. So in westernized populations, we've routinely seen that these species are potentially a different species than in non-westernized populations. It really depends on diet. People that on different diets, these species are completely different and that are popping up. So this is really a ongoing debate in the field, right? So how many species are we talking about? So in earlier years, we've really linked something called diversity to these diseases. So if there's so diversity is the different number of species. So if the diversity sort of drops, so there's the you have, for example, 10 different species, but another person might have only 3 or 4 different species, then that's a lower diversity, right? So we've previously sort of linked that to different types of diseases, but nowadays we also know that the picture is not that clear. It's not just diversity; it's also which species are there and to which numbers and what they are doing. So yeah, that's more or less when we talk about the number of species. The oral microbiome is in terms of how many different species there are, but less. And it also is a bit more transient. And like I mentioned, the oral microbiome is in different sites is also quite interesting. It's very different. So in the saliva, it's different, for example, than what is on the tongue, what is on the teeth. So they form very specific niche pockets where they perform different functions. So that also changes quite a bit. But in terms of the number of different species, it's a bit less than in the gut. Or that's at least what we found in our study and in other metagenomic studies. Yeah.
How about translocation?
Sorry, translocation.
No,
That
no,
was
please.
the second part of the question. Yeah. So
I
the
shouldn't have given you 2 questions in one go.
it's fine. We get carried away. So in terms of translocation, I mean that's also still something that is under study, not just by us but by other people as well. So because I mean, at least physiologically from the mouth down to the gut, it's all connected through the esophagus, through the stomach into the gut. So that is connected in that way. So that is the hypothesis that these bugs can translocate in such a way, right? That they go literally from the mouth to the gut. So that is the hypothesis, not necessarily through the bloodstream, as far as I know, yeah, that they actually go through the gastrointestinal tract and then come up in the gut. So I might be wrong, but I'm not necessarily familiar with studies that have really shown this, right? So exactly how that happens. But it's not just there's a couple of groups around the world that have really shown that this tropism is we call it tropism, so gut oral tropism is a phenomenon of a lot of different diseases when it comes to the microbiome. So especially in terms of chronic diseases and things like that.
I'm worried that this next question is going to lead us down a rabbit hole, so we can keep this short because I want to get to the findings, but purely because I'm interested in, let's say, development. Has many groups sort of taken, I hope someone's done this, but taken samples at, let's say, different stages through childhood to puberty to adulthood and sequenced the saliva or the oral and gut microbiome to see if that changes, let's say, across development. Obviously, there's other variables to take into factor, like the environment, the ecology that we're in, your diet and everything. Has anyone done that?
Yeah, so there's actually been a lot of papers in the last while around that across that development that you're talking about. So how from very early on, so from infancy, so from early babies on through adolescence and that whole change in the microbiome. So there's been a lot of work around that, and we, like you said, don't go down a rabbit hole with this, but that there is very, very interesting work around that early sort of colonization as well. So again, not if we don't go into the details, but a lot of work around, for example, different feeding strategies. So let's say, for example, breastfeeding versus non-breastfeeding, these kind of things, how that impacts the oral microbiome or have different outcomes, all these different things in early sort of colonization. So in just after birth, how those early colonizers, what are they actually, right? So what are these species that are initially found within the gut microbiome, and then how quickly do that change over time, and what is the impact of that, right? So that's neither, that which one is good or bad or these kind of things. It's just there's been a lot of work around change in development.
That's so cool. That's so cool. It's almost like microbial sociology, let's say. It's just
Yeah,
sort of trying to understand these
yeah, there's
different.
been there's I was recently at a conference in Embel in Heidelberg, and there's a lot of work around these changes in the microbiome early on, and that has how it might, again, very associative, but impact sort of some childhood diseases, but also then later on, also in terms of cognition. So there's a lot of potential similarities as well between some of these early things, and then also again in the neuro diseases. So some of these species, so these early diseases in terms of childhood, so things like autism or something like that, and then also later on in the neuro diseases, very similar markers in terms of the microbiome. Again, very much open still and to do, but yeah, very interesting.
We can probably delve into that later because about how your work can be sort of expanded
Yeah,
into different areas. But let's bring it back home. And if you were to give me the TLDR, so like a 1 or 2 sentence about, let's say, the main finding of your paper, what is it, and what's the most sort of important takeaway from that piece of work?
yeah. Also, always very very difficult to do that. Like you're very well familiar with. Yeah. So I think that the main takeaway that we found is the existence of oral gut brain axis that influences Parkinson's disease and cognitive decline. So that, I would say, was the main takeaway, is that existence of an oral gut brain axis, and then that this goes hand in hand with virulence, which we can touch upon now. So what that virulence mean? But I think that's the main takeaway, is that existence of oral gut brain axis that influences cognitive decline in Parkinson's. So that's the major, which is almost probably, I would say, the title of the paper, but that that's what's basically the main finding.
Yeah, I mean, no, that's good because it's a nice baseline that we can start off that we both clearly understand because you know more than me, obviously. So let's take it up a notch. When you compare the microbiome across the groups, so from healthy control to mild impairment, to Parkinson's disease, and then Parkinson's disease dementia, I mean, obviously there's going to be a lot of changes, but what were the sort of the big global differences, let's say?
Yeah, that's also, I mean, it's actually a good question. We can, like I said, dig a bit into the details about this as well. So we, again, us and others have also shown previously that these major changes that we associate sometimes with disease, like these major drops in diversity, like I mentioned, which is something that we've always thought is a very good marker. So what we call a richness or diversity is not always happening in Parkinson's, that you don't necessarily see compared to healthy controls, that we see this drop in the number of species that are there. That is also something that we found. We don't really see that drop necessarily in the oral microbiome. We did see that. So we did see. So these are the global changes, sort of, that in the oral microbiome we do see that the number of species from the healthy control to the dementia patients do drop slightly. So that is the one major finding in the oral microbiome that we found, but again, not extremely compelling. And then it really came down again to the individual species that are actually changing. So there's a couple of species that have previously been associated with Parkinson's. So these included things like Bifidobacterium and E. coli as well, but Bifidobacterium primarily is something that we've seen enriched usually in Parkinson's patients. So what we found was, again, these species that are enriched in our cohort as well, but that this do change slightly over the spectrum of cognitive impairment. So where previous studies have really just compared healthy controls versus Parkinson's patients, so not really looking at that stratification of the cognitive impairment, we saw that a lot of these Bifidobacterium species that we've known are enriched only come up when they're on the dementia stage. So they not necessarily come up in the mild cognitive impairment stage. So they're still sort of at the same level. And then in the dementia state, that's only then when these species gets enriched. So it is as a consequence of cognition potentially, or at least a later stage of Parkinson's, that these species then start to get enriched, which is a known sort of microbial marker for Parkinson. So that is some of these changes that we found. And then in general, again, a bit more broader, what we found is that we've seen the sort of in enrichment of these types of species, but then at the same time in the dementia patients, a depletion of what we would call, in the correct term for that is commensal bacteria, which is more associated with sort of a healthy gut microbiome. So we actually see in the dementia patients that they deplete, so they go down. So these are in quite often we think of them about, or when you look at their functions, is a bit more sort of butyrate producers. So butyrate has an important role in cognition as well. So butyrate goes into the bloodstream, and then it has very specific functions in neuroprotection and these kind of things. And the microbes produce a lot of butyrate. That the microbes produce some of the most butyrate in the body, right? So they're responsible for the highest production of butyrate. And these butyrate producers actually drop in the dementia stage in terms of their abundances. So those are sort of the there's a bit more detail on the major findings that we found. Yeah. The first thing, basically, if I want to summarize, is that these microbial changes do happen across, different changes happen across a mild cognitive impairment and dementia. And then the second thing is sort of that we do see that there's depletion in dementia of butyrate producers and more of an enrichment of more potential pathogenic species, right? That do go up. Yeah. So that's more of the detailed changes. Yeah.
So apologies if I'm repeating, but why
No,
not?
of
Because
course.
I'm learning. I would say there would, let's say, three global changes, right? So number one would be a decrease in diversity, specifically between the mild impairment and the dementia patients. And then secondly, there would just be an increase of, let's say, pathological microbes as well.
Pathogenic microbes. Yeah.
Pathogenic, sorry, not pathological. It's pathological liars. We're not going to be describing microbes as pathological liars. And then the third one would be the decrease in, let's say, protective or commensal microbes. Now, I mean, I understand because you're a bioinformatician, so I might be pushing you a bit on this with this question. But so you mentioned the microbes that could release butyrate, and are there sort of, I guess what I'm trying to get across is, do we know that we have all of these different species, but what do we know what all of them do? Like, do they release short chain fatty acids? Do they help with, let's say, dopamine transporter receptor expression? Like, do we know like what's going on?
That's a very good question, because some of the work that we try to do as well is really not go beyond the mere presence or absence of these species, right? So not just us, a lot of other groups around the world as well. But from a computational side, we try to do that, to move beyond that. So some of them we do know a lot of their functions, because they are, some of them are grown in the lab. We know some of their sort of general functions, what they produce, sort of if they are more pathogenic, if they are less pathogenic. And we also know that's a bit from the history, right? So where they lie in terms of their taxonomy, right? So with these more low abundant species are sometimes a bit more pathogenic. They're not that abundant. And then in diseases, they become a bit more. But in terms of their functions, some of them we know, some of them we don't, right? So we know some of them. For example, when we talk about the butyrate, some of them we know are known butyrate producers, right? So that we know through different reasons, because of microbiology, but also through some of the functional studies, et cetera, et cetera. We know that they might be potentially butyrate producers. So these included things like Faeculobacterium, Roseburia, Blauschia, these kind of things. These species, we know they are known to be butyrate producers. When it comes to the functions of these species, what we also try to do is we try to move beyond the mere presence and absence of them. So computationally, we can look at their gene content, right? So we look at the gene content of these species, and then without going into the detail about the bioinformatic approaches, but we then try to see, we compare this against databases, these genes, and then see what their functions might be, right? So then we can go back and see, do we see, for example, some genes that are more in, let's say, the Parkinson's patients, that they have certain metabolic functions, right? So we do some fancy statistics, and then we say we have a bunch of genes, let's say in the the dementia patients, that are enriched for certain metabolic pathways because we know these genes have a particular function. This is not a complete reconstruction necessarily of the whole metabolic pathway, but it at least gives us some sense of what then these species in this community might be doing, which is partially what we've done as well, right? So we did try to look at these methods. So that's the one way computationally we do that. And then the second thing that we do as well, which is a bit more sophisticated in essence, which we didn't do in this particular paper, but we also reconstruct genome scale metabolic models, which is a lot of what my previous work has been on as well. Is so we reconstruct sort of the entire metabolic pathways computationally, and then we simulate metabolic flux in silico. So we reconstruct the whole metabolic network, and then we sort of see what they can produce and what they can't produce, right? So by simulating that metabolism computationally, which again is not what we did here, but that's one of the ways we try to bring, to answer your question, more just from what there is to what they're actually doing. Because in the field as a whole, there's a lot more work showing now that it doesn't really depend on what species are there. It's really what they are doing. But that's the key, right? So because in one instance, that species might be doing nothing, and then in another environment they might be doing something completely different. So the functions is the important thing. And across diseases as well, a lot of studies have shown that the functions remain. The species might change up and down, but the functions remain. So what they are doing, so if you see, for example, this depletion of butyrate, not necessarily the species, that's the thing that might change. Or what the functions do is the important thing. Yeah.
So basically, if you understand the metabolism of each, let's say the metabolic function of each microbes, then you can sort of predict what they can release. Is that
Yeah. So that's
okay?
more or less what we can do. Computationally, we can predict that, right? So we can sort of say, so we've reconstructed some of these metabolic models that I mentioned for different organisms, and we've done this for metagenome, so in the context of metagenomics, but also in individual species. And then you can predict sort of, okay, these species can produce certain things like butyrate or acetate, some short-chain fatty acids. Some other ones they might actually be able to release, I don't know, some amino acids or something that might affect host physiology or whatever. So that's remains a prediction, but we can, like I said, we try to computationally at least get closer to functions than merely what is there and what is not. So if you're just looking what is there and what is not, those are good markers, right? So they might be good for something along the lines of diagnostic biomarkers or these things, but not what they are doing is actually the important thing. Yeah.
I guess there's 2 points I want to get across before we move on to the definition of virulence, because whilst we can use machine learning to have a better understanding of, let's say, the association and the correlation between, let's say, certain microbial species and any sort of clinical phenotype, if we don't understand the function and the mechanism, we can't limit the noise and focus on specific species, let's say. Which then moves me to my next point with regards to the sort of release of short chain fatty acids or SFSAs like butyrate or acetate. Can you validate them, let's say, clinically via like saliva proteomics or not clinically, but in
Yeah.
person with saliva proteomics or any of that? So you're then sort of targeting it through both ways. You have this sort of prediction in silico computationally, but then you're also coming in from the other end from, let's say, a top-down perspective.
Exactly. Yeah, that's exactly what we try to do with multiomics in that sense, right? So a lot of the other work that we're doing at the moment is more around multiomics. So where we would do these things from the metagenomic side and do some of these simulations, look at the species, what is there. But at the same time, we do some metabolomics, for example, and then we can see, do we actually see a change in that? Again, it's always difficult. It might not be what we predict from the metagenomic side that it is exactly these species, but then you can at least have an estimation. Okay, now we see more butyrate or more acetate either in the feces or in the blood, actually in certain conditions. That is exactly what you can do, is to try and validate it in that sense, which is not always a direct validation of, let's say we find a couple of species that we think are the butyrate producers, whether or not it's really them. For those types of things, you really need to go back to more tracer kind of experiments, which you kind of label, for example, some precursors and then see if these organisms can do that. So then you do a very fancy sort of mass spectrometry and things. You can trace metabolism experimentally and see what is being produced, which is still in it is a lot more difficult, and especially to do it in humans, right? So, or in animal models and things, it's possible, but it's more difficult to trace all these pathways, right? But we tend to do in terms of multi-omics, some metabolomics as well, and then see, do we actually see these changes, or at least if we are not 100% sure, do we see some precursors or something around that type of metabolism? Because it's not just short chain fatty acids. A lot of sort of tryptophan metabolism is involved in the bug. So they produce a lot of these metabolites, intermediates of tryptophan metabolism, these kind of things. So there's a lot of things we look at in terms of metabolites, because they're metabolically, the metabolism is still a major part of these bacteria, apart from the virulence that we can get to. But the metabolism is one of their major functions, right? So how they process a major part of processing nutrients from the diet as well, right? The nutrients that reach your liver and things is first metabolized by the microbes, right? They first go through the stomach, then they reach the gut, and then that goes obviously to the nutrients, then go to the other parts of the body. So they major metabolizers in the gut.
Do you? God, I've got so many questions, but hey, do you factor that in when you model? For instance, the response. Let's say, let's break this down. Let's say in vitro, even though you can't grow all the microbial species, but let's just say for hypothetical sakes, you have the gut microbiome and then you have the oral microbiome, and the oral microbiome can sort of respond. Both of these can respond to, let's say, nutrients, right? In vitro. Can you predict, let's say in silico, that maybe the response will be different because the nutrients, the oral microbiome would respond to the nutrients first, and then that can then have a knock-on effect on like metabolically, and so then the byproducts of, let's say, that metabolism would then be interacting with the gut microbiome, if that makes sense. I don't know.
Yeah, no, it makes complete sense. It's something in the field of this. We're now going a bit more towards this metabolic modeling, which is something I've also spent a lot of time on in my research. And these are all things you can consider to a certain extent, right? Because it depends on what you can measure and what you make assumptions with, right? So any type of modeling, especially in this context, do make a lot of assumptions, right? So, and again, it depends on how many assumptions you are willing to make and can make, and then how much you can measure. So in that context, if you collect enough data, so in theory, those things, people have done this and it is possible, right? So if you do, for example, look at this in silico modeling, but you also do metabolomics, then it's always a question of, are these metabolites being consumed or produced? But you can make some assumptions there, and then you can see, okay, what, for example, is again in the gut, what is there, and then from metabolomics combined with in silico modeling, sort of what do they then produce, given they've already went through some sort of metabolism when they get there. If you have the different parts, measure the different parts, these things are possible. So at least in theory, I'm also talking now sort of just in theory, because we haven't necessarily done this, but these are all assumptions. So, and that the assumptions get even more when you go to, for example, we've done some work on host metabolic modeling as well, where we model liver metabolism, for example. There you can obviously do transcriptomics and metabolomics and a lot of different things to sort of see what metabolic pathways are active and these kind of things. But even there, when you talk about the input into these models, you make even bigger assumptions, right? Because you don't really know what are these metabolites. A lot of the metabolites we can't even capture with experimental analytical techniques, right? So the vast majority of metabolites we don't capture. So then you make even more assumptions. So a lot of these host modeling, we then actually give just a normal diet, and then we say, okay, we will. That's the best assumption we can make, right? But in theory, what you're saying is exactly what we try to model. So there are some research done around exactly that thing, especially, for example, even in the gastrointestinal tract, right? A lot make these assumptions, but going from the different sort of the small intestine to the large intestine, making it like a pipeline, right? So try to model this, the one goes to the other. So there are people that try to do this. Again, it depends on what you can measure and how much assumptions you make.
I mean, the reason I ask this is like from a translational perspective. Like when I was working in VC's venture capital, like the one thing that we sort of got ingrained into us as, let's say, interns or associates is that the model system that you use to test your disease model or to test a drug or understand a signaling pathway, therapeutic target has to be translatable as much to, let's say, clinically. So we can theorize and say, talk about all multiomics and everything in theory, but I know it's very difficult in reality. Like having that ability to try and replicate how the microbiome can influence even how you take a drug or, the
Yeah,
environment and everything would be really cool. Yeah, because, I know it's difficult, but I think
yeah.
it would be,
Essence when it comes to these, you're 100% right. So these things should be translatable, and a lot of these metabolic models that we build, I think, gets closer to the translation already, because we don't we are doing things in the gut as well that we only have a proxy of, right? So it's not something else that you work on a certain cell lines, whatever, you know exactly what is happening, you can measure everything. We work with a proxy, right? So we kind of say, okay, even the feces that we use as to take these DNA out of, right, and say these are the species that are there, is not necessarily representative of what is happening higher up in the intestine, right? So the feces is a proxy of the microbes that are there. We know that it is a very good representation. I just mean it's always some assumptions that you make, or some drawbacks to what you measure, but getting it as close as possible to translation, you're 100% right. So I think these, for example, the metabolic models, we try, they get a bit closer to some sort of translation, because we can even try to model some drug perturbations to see, for example, with these models, is if a certain drug, how can they metabolize a certain drug, right? So that, for example, you take a drug, in some people, this is a major sort of area at the moment, sort of this interplay between pharmacology and microbiome, right? So certain drugs work in certain people, and certain people it doesn't work, right? So whether or not the microbiome might play a role there, that they can metabolize that drug to another compound, which is then not as effective as that drug, right? So in certain patients, that drug doesn't have an effect, which we previously didn't know, and it might be the microbiome, and that's what we try to sometimes do with this modeling, right? That it can actually take such a compound, metabolize it to some other compound, and then that's why it's not effective. So again, I don't know all the details, but these are some things that are going on in the field as well that might explain some of these phenomena. And I mean, we are aware, obviously, that, for example, that the cancer drugs and things like chemotherapy and immunotherapy are largely influenced by the composition of the microbiome, right? So that is, a lot of studies have shown this, that the microbiome composition in certain patients that does help, and things like anti-PD-1 and all these things, they are affected by the microbiome.
that's really cool, because I guess, like, from, let's say, a drug's perspective, you have PKD
Yeah,
modeling at the moment, which sort of,
yeah,
I think, looks at drug efficacy and metabolism as it sort of goes through the body. But and then you can use predictive models to look at toxicity. For instance, like I was at the toxicology unit for my PhD, but I didn't think really about how you could maybe sort of use modeling and metabolomics to try and see how the microbiome can affect, let's say, the toxicity or the efficacy of a drug. Which, whoa, that's
yeah.
cool. But I think we should stop there, because I'm going to go down another rabbit hole. So
Also, I
what?
think we should stop there, because I'm talking from something that I haven't done before, something that I know is going on. So I might also not
Yeah.
say 100 things, but then I might also land into trouble if I say that the microbiome is the be all and end all of everything, and it is not.
I mean, everyone's biased about their field of study, but this is all hypothetical, right? So one more question before we get onto virulence. I apologize. So because I worked on, for brief amount of time, a rare disease called primary ciliary dyskinesia, which is basically a rare disease that affects the cilia, which are these sort of tiny little antennas located on every cell, and they play a big role in signaling to also removal of debris, cellular debris, because there's two different types of cilia. But anyway, in PCD, one of the biomarkers that they use is the release of nitric oxide, which is really cool. And they did some mechanistic studies to sort of understand why. Could This is a big leap, but given like the release of these short chain fatty acids and all of these sort of amino acids and metabolic, could that have potentially effect on your breath? And has anyone like let's say experimentally, would that be potentially a line to go down, or is that
Yeah.
too broad?
No, it definitely does, right? So again, not something that I'm 100% directly involved in, but in terms of the oral microbiome, there's been a lot of work around that metabolism, especially things like uh, nitrate and nitrite metabolism in general in the oral microbiome, and how that affects things like taste, and in obviously affects things like breath smells and these kind of things. So there has been a lot of work around that, so especially nitrates and nitrite metabolism in the mouth, something that is very much actively studied in terms of the microbiome, and together with things like metabolomics and things like that, and how that affects different functions, but also things like that. Yeah, most definitely. Again, with that, the details, I won't be able to know the exact mechanisms of these things. But
So now let's, after a very sort of long hiatus,
could be a good detail.
let's go. So the three changes you saw were decrease in oral diversity, increase in pathogenic microbes, and decrease in, let's say, neuroprotective or protective microbes. You talked about, I think one of your most novel findings was the fact that you had oral microbes and virulence factors appearing in the gut. Yeah, talk to me about that. What exactly is going on there?
Yeah, 100%. So just again, sort of definitions in general. Virulence is obviously a kind of bacterial virulence is a bit of a broader term, but in essence refers to a pathogen's ability to cause disease or damage or even death to a host, but it's a bacterial pathogen's ability to cause the disease. And it represents a sort of a degree of pathogenicity which is largely determined by virulence factors, right? So these virulence factors are things like toxins or enzymes or things that help this organism to be virulent, right? So, and we associate this in general with pathogenic bacteria, right? So that's the other concept that's important, which, I mean, all bacteria are not bad, right? So all bacteria are not virulent. So as we've just talked about a bit, like a lot of these microbes in your gut help you digest food and everything. They commensal, they much in symbiosis with the host, they don't cause disease or whatever, but there's a proportion of these organisms that are sort of more pathogenic, and there's a major ecological thing that they are sort of in low abundances, and then sometimes when they get higher in abundances, then they can sort of cause disease, right? And these are the factors that allow this are quite often virulence factor, right? So they might be very specific genes that do certain things. So they might allow the secretion of a certain toxin. They might allow, for example, adhesion to the gut wall, where they can form a biofilm, and then they can release it when these when they cause infection, for example. Certain certain bacteria causes infection. These virulence factors help them to be able to cause that infection. So when they adhere, for example, to a certain tissue, and then they do certain functions, and they can be pathogenic. So these are the virulence factors we refer to, and we can at least sort of relatively, but we can quantify these virulence factors using metagenomics, right? So like I mentioned, we take all the DNA. In the one hand, we look at which species they are, but at the other hand, we look at these genes and what they are doing. So we can, part of that is metabolism, which we just discussed, but virulence is a major part of it. So we can, to a relative extent, quantify these virulence factors and then see what are these virulence factors, and are there more or less of them in certain conditions, and what might they potentially be doing. So what we've done in this case is we've actually found, or not what we've done, what we've found is that there's this translocation. In essence, we pick up a lot of oral-like genes in the gut. So when we sort of did the metagenomics, we found some oral genes we normally find in the oral sort of microbiome, and these genes have a large proportion of them are virulence factors. So there's a statistical enrichment in Parkinson's for these virulence factors. So in the healthy controls, you find a lower amount of these virulence factors, which are oral-like virulence factors, in the lower amounts in the healthy control versus the Parkinson's patients. So in the Parkinson's, we see this enrichment of oral-like virulence factors. So these genes that might be causing some sort of pathogenicity. So that's the first thing that we sort of found, is just as we call them enriched virulence factors in Parkinson's. Right? These virulence factors are more. So that was the first finding in terms of the virulence that we found. And like I said, this is a major part of bacterial ecology in that sense. And then we did a bit of digging into these virulence factors, and we did again a bunch of sort of fancy computational things, and we looked at some correlations of them, etc. So with the species, and then the hypothesis that we sort of came up with, because of a lot of these more network type of analysis, is that we see, whilst we see this enrichment of more pathogenic species in the gut, we see that they are very highly correlated with these virulence factors, whereas the more healthy bacteria that we talked about, the butyrate producers, are negatively correlated with them. So they kind of go down. So you kind of see as these pathogens go up, we see more virulence factors. And then at the same time, we see a drop in these commensal healthy bacteria. So that we hypothesize that there might be some sort of competition going on there, which the virulence factors might mediate that in a way that they push down these other bacteria, and at the same time these more pathogenic bacteria come up. And this is again not measured. This is a more of a hypothesis that we state. We've measured this, at least that these virulence factors are higher, but the mechanism sort of that is something that we state as a hypothesis at the end, that might be what is happening. So you kind of see this virulence factor release. Some of them we hypothesize might go into the bloodstream because some of them that we picked up are known to be able to bind, for example, to receptors on the brain. Some studies in vitro have shown that some of these virulence factors can do that. They can bind to brain receptors and things. So that's the hypothesis that we make, that they might go into the circulation, they might do certain things, and that's a potential mechanism that we're following up with now to see if we can somehow validate that. Yeah. So that's virulence in general. I know that was a lot, but
no. I'm gonna throw some points at you just randomly in no order, and then we can go through it. On your last point with regards to virulence factors and receptors, that sort of got me going. Can you sort of then, let's say, grow sort of neurons or iPSCs in vitro and get these virulent factors and just add them and see what happens?
That's what we want to follow up with, right? So that's like I said, these are we try to at the end sort of narrow this down, because we're working with hundreds of genes, right? And so we found these virulences, but then we try to look at their specific functions and sort of narrow them down to specific ones that some studies have shown, okay, what they might be doing. So then that's where we come up with some of them that studies have shown that they can bind to some of these brain receptors. So experimentally, that's what we want to follow up with, right? So you grow them, you put this in virulence factors there, then you can measure, for example, which pathways are some of specific pathways activated in these cells. Then if they bind, what these pathways, what is it, particular signaling pathways or whatever that might be responsible for other downstream processes. Then you can go get closer and closer to a mechanism, and which, again, with the broader philosophy that I talked about earlier on functions and not species, that's where you can get to a real function of these microbes in the gut and exactly what they do, which then can eventually be something that is easier targeted, certain functions like that. But those are the kind of experiments that we want to, and also some mice experiments potentially to see what these virulence factors might do. Yeah.
I mean, just because I'm moving into protein biochemistry now, so it'd be really cool to. I don't know if this is possible, but hey, if you could purify, let's say, receptors, and then purify the virulent factors, and we'll just chuck them in and see if they have bind, and see, there's an assay called microscale thermophoresis, which is called MST, and what you can do is you purify proteins that you think are going to bind with each other. One of them's tagged with, let's say, a GFP tag or MNeon tag, and you basically put them together, see in Brownian motion, and then heat them up and see how they sort of bind with each other, and then you can measure sort of the binding efficiency. Yeah, I don't know if people have done or have even considered doing that with virulence factors. I don't know if you can purify virulent factors, but
some of these, I mean, I don't.
Yeah, but we're out of that territory.
But some of these virulence factors, I mean, they are proteins, right? So they are molecules and whatever. So it's possible. Some of the more known virulence factors, it is possible to don't even have to purify them. Some of them I know you are available to even buy because they are factors that are known. It's certain compounds that are known. So you'll be able to get or purify them in that sense. Make sure that they'll produce and then purify them and do such kind of experiments, which is, like I said, exactly what the follow up is in our minds, at least, is to do these kind of experiments. Yeah.
You asked me this last year, I wouldn't have told you anything about protein purification because it wasn't in my wheelhouse. Anyway, so back to virulence factors. Like, if I got this correctly, you can have, let's say, in 2 patients, the same all microbial species, but the containment of virulence factors can be different within that individual species between patients. Is that correct, or am I
Um,
missing something?
that is correct. 100% necessarily what we've found and what we've measured in the study. So we more looked at, again, not individual groups of patients. So again, going from mild cognitive impairment to dementia, in those two categories, we found that there is a very sort of linear increase in these virulence factors overall in the whole community. So not necessarily in individual microbes. So when you look at the healthy control, then to the mild cognitive impairment, then to dementia, you see this linear sort of very stark increase in the number of virulence factors that are there in the gut. These are again, technical thing, but I mean we found them by looking at specifically oral genes. So that's why we hypothesized that they are oral, could come from the oral cavity. But it's not so. But even to your point, I mean, it is possible that within a single individual that these things differ as well, between different sites and things. But that's not exactly what we've measured. But yeah, so we do see this increase, and then we sort of, as I said, digged into these virulence factors, and then found very specific ones that are also significantly increased in their literally their abundance as well, and then sort of found them to have very specific functions that might be interesting.
As in, these virulent factors can have, let's say, indirect effect on diversity
Yeah,
by.
that's sort of what we again very broadly sort of show in a way, is that it has an effect on the microbial composition in the gut, in the sense that these are correlative measurements that we make, but we see basically an increase in the virulence factors and an increase in pathogenic species, and then at the same time a decrease in more healthy butyrate producers commensal bacteria, right? So these three things sort of happen at the same time, right? So then we sort of, it's a bit of network analysis that we did show that we think that these virulence factors might mediate that, right? So the virulence factors might push down these more healthy bugs in the community by producing potentially some of these virulences, so they outcompete in a way the more healthy bacteria, which then goes down. So you kind of then get to what we call a dysbiosis stage, right? So this is the whole concept of dysbiosis, that you see this changes in the microbiome that puts it in an unstable state.
Okay, so I'd say from this, we sort of have 5 key points to take away from this. The fact that there's a reduction in diversity within the oral and the gut microbiome. Um, there's an increase in, let's say, pathogenic.
Really.
I think my subconscious mind is saying something to me, but the decrease in neuroprotective or protective species, the translocation of, is it just the translocation of oral microbes to the gut, or is it like virulent factors specifically which are being translocated?
Translocation of oral microbes potentially. We specifically look at the virulence factors, but it's a translocation of oral microbes.
And then we have the increase in virulent factors. Those are, I would say, like, are the 5 key sort
So
of. So let's, with regards to, and then, we've discussed how we can then dig in, let's say, mechanistically to sort of understand the causative mechanisms of what's going on, and add, let's say, multimodal omics as well, which, was a very sort of fun thought experiment. Like, how could this be? The one thing that really annoys me about the modern world is sort of, I shouldn't get too aggy about it, but the promotion of all these prebiotics, these probiotics, and keep your microbe healthy and all of this, which, to be frank, I think is very annoying unless it's not from, let's say, a biotech or health tech company who wants to
yeah,
go
yeah.
down the FDA. Like, if you're selling this to Holland and Barrett, you're selling a lie, I think. And that's my own personal opinion, purely because I think the science is still not there, and there's still so much complexity to be understood. But do you think, with the machine learning you've done and your ability to predict, let's say, cognitive impairment based on, let's say, these 5 variables, could it be possible to, let's say, work with biotech or the pharmaceutical industry to try and actually develop evidence-based biomarkers or panels, or evidence-based prebiotics and probiotics? Because one of the, let's say, selling points from these companies which annoy me that are selling these probiotics or prebiotics is the fact that you can maintain diversity of the microbiome, which is a very flat and simplistic claim. However, if you were to dig into it as a scientist and sort of understand that mechanism and collaborate with others who don't want to scam people, you could potentially be onto something.
Yeah, I 100% agree with you in that sense. I think I have the same sort of opinion as you about probiotics and prebiotics, and I think a lot of people in the field do have that opinion in the microbiome field. I think others don't, maybe, but what you called non-evidence based alterations of the microbiome, because there's a lot of so the normal probiotics, prebiotics, and again, I'm not talking about people that try to use things like live biotherapeutics, which is not really that. I mean, there's evidence based and very specific consortia of bacteria in the context of a disease. I mean, the trials that we've seen is promising in that sense, which is different. I mean, there's in this past week trial or two, the evidence that came out, which is really promising results for different types of diseases. But that's different, right? So there's this
Biotech. That's
that's
biotech. Yeah.
biotech in the sense that where they also very similar to what we've done in academic research, but went down the route of looking more in depth, right? So saying we have a consortium of people, we sort of see what are the changes in those microbes, find very specific microbes through different fancy bioinformatic machine learning algorithms, etc. Then we find a consortium of bacteria that we can with good confidence we can grow them, we can put them into some sort of therapeutic, and then live biotherapeutics given to the patient. And those results have been promising throughout, if it's done in that way. The other side of it, of just putting, random thousands of I don't know, lactobacillus into a pill, and then that there's very little evidence. So the evidence that are there for that is again not 100% where I come from, but where they is some instances where these can sometimes be useful in certain cases where they use this as a adjuvant therapy alongside something else. Then you might see slightly better effects, whatever. But the evidence is still very much up in the air whether or not this works. So I do agree with you that it is one of the major tricks of that field in the last while, that they think that everything is to do with the gut. So everyone just munches a bunch of probiotic pills and and eats a lot of, I don't know, fiber, and then everything would be better, which is not the case. So in again, this goes back a bit more to the functions that I talked earlier, is that it's not about the species. The functions remain, right? So even across diseases, what we found in some of our other work in the group is that even in the context of different diseases, the changes that we see in the microbiome, it's different species in different diseases, but the functions remain, right? So the types of functions that are changing in that dysbiotic state. So you have, let's say, I don't know, liver disease and Parkinson's, which are two different types of diseases, but the species that we see that are changing is different, but the functions are very much more similar. So the functions of those species, so what they might be doing, is very much more similar. So that one goes back to the metabolism, but also the virulence and these things. So I do see value, like you say, in going down this route of really understanding such a mechanism to be able to eventually target this, right? So, or like you say, even use it as a proper biomarker, right? Because that was the other thing in the field that I think people thought we're going to be much more easily be able to use the microbiome as a biomarker, just the species, which is more difficult than we think because it's so transient, right? So it's not, I wouldn't say it's impossible, but it changes quickly. It's not necessarily that robust. So again, we need to go deeper to understand it better. Then we can come up with either targeted therapies or targeted diagnostics, which is a lot closer to the reality than just looking, okay, you have more of the species, so you are you're going to develop this disease. It's not that easy.
Yeah. There's 2 points I want to say to this, because you touched on something which is really important, which is the distinction between, let's say, presence and function.
Yeah.
Just because there's a presence of, let's say, a certain species in a disease population compared to control doesn't necessarily mean that that has a contribution to the disease, because we don't know what the function is, right? The second to that is, at the same time, I'm not an expert, but one thing when, I look at biotech companies developing prebiotics or probiotics, and something that you mentioned with regards to biomarkers is the transient sort of dynamics of these sort of communities. So do you think maybe that it's sort of sifting through the noise and being able to focus on, let's say, the fundamental key differences between, let's say, a mildly cognitive impaired patient to a dementia patient to a healthy control, and understanding that sort of function to truly develop a therapeutic or a biomarker?
Yes, 100%. That's what I think it is, right? It's the the association is easy, right? So making the association between, especially with something that's as dynamic as the microbiome is, right? So making an association between a certain disease condition and then changes in the microbiome because of that disease that's the relatively easy part, right? So we've now shown it in a range of different diseases, and in certain diseases we know that signatures are much stronger. So in certain conditions, so something like neurodegenerative diseases, there's a very strong link between the microbiome and the brain. So between the microbiome and the condition, there are these types of diseases where you find it. Other conditions you might find it, but the strong signal is not that strong. But in both cases, you really, I think, need to dig down to more of these functions and understand that mechanism better before you are able to potentially translate that. And I do think this goes hand in hand with more, not just omics driven things, but also experiment, right? So going back and actually testing this in vitro and in vivo and see whether or not you can recapitulate some of these findings. So for example, in the case of the virulence factors, go back and see, okay, if we do the correct experiments, can we show that there's a mechanism that exists here that holds true in vitro and in vivo? Then you can go maybe design a better therapeutic for this but apart from that, I think things like live biotherapeutics and FMT or all those things in that sense is working, because I mean the hypothesis of the live biotherapeutics is a bit different, but FMT is basically restoring that balance. So there is a lot of evidence by saying, okay, you have this dysbiosis in your gut, so you have the species changing, and that even though you don't know the mechanism, by restoring that balance, you might have some positive effects. So I mean, FMT has worked well in things like C. diff infection and these things. So because that restores that balance in the gut, that's basically what it does. Although there are some variable arguments about that, saying that FMT might rather be a tool to study these things rather than actually becoming a widespread therapy, because it's quite difficult as well to use it as a therapy. So it might become more of a tool to say, okay, or a technology to say, if we do an FMT, what is the consequence? Do we restore that balance? Do we not restore that balance, etc.? Because, for example, even in C. diff, there's a lot of relapse still after FMT. But to go down to a very specific microbiome-based therapeutic, I agree that you kind of have to go to the functions, right? To see really what is happening. Then you can be able to target that in a different way. So let's say, in this case, for example, it is a specific virulence factor, and there can be ways that you can target that virulence factor or that specific microbe in one way or the other that produces that virulence factor in that condition. Then you might not be 100% relieving the symptoms, but it might be helpful in one way or the other in this case. Yeah.
I mean, one of the things I'm really passionate about is this translation, right? And being
Yeah.
able to see how we can translate our findings into a diagnostic or therapeutic.
Yeah, me too, actually. Yeah.
Yeah, and I think, but also as someone who's trained as a mechanistic cell biologist, and also from an investment perspective, I think if you have a better understanding of the mechanism, you can de-risk the biology. And so you have much more confidence in the therapeutic or the diagnostic that you are spitting out and producing. So, we discussed this, but what would you say is the next step in summary to really get through the noise and through the weeds to try and hone in and focus on maybe a specific population or a specific virulent factor, and sort of hone in just first Parkinson's disease, because I think it's going to be vastly heterogeneous between different
Of course, yeah.
diseases. This sort of notion that your oral microbiome is completely linked to just your cognition as an umbrella term that we see in the modern world is, to be frank, I think insulting to my intelligence or potential intelligence, because I just think it's too
No, no,
broad.
yeah.
But
Yeah.
yeah,
Oh, I agree with those broad terms, of course. I mean, I don't think that is when it comes to the translational side of things, or I mean, also quite interested in translating these findings and also about the biotech world in general. And I do think those very broad, more consumer based sort of companies is not doing the field a favor, I think, because in the long run it might work out, might not work out, right? So and that's I mean very broad things that you can, like I mentioned earlier, you just have to eat a lot of probiotics and fibers, and then you're going to be fine. These kind of assumptions, or whatever you want to call, like you say, oral microbiome, and just take care of your oral microbiome, and then everything's going to be fine. I mean, I think it's good that there's been with the new studies and stuff in terms of oral microbiome light being shed on these things. So, for example, in our case, we also put at the end of our paper that, I mean, these are things, for example, oral health and things might be things to be considered in this case, right? So, for example, in the case of Parkinson's patients, they didn't, in our study, they didn't explicitly, in the clinical arm of it, they didn't explicitly look at oral health, right? So, for example, if these patients have a good oral health, do they actually brush their teeth regularly? These kind of things that we don't know that, but there's something that we, in one of the, again, I'm not a clinician, but in one of the questionnaires of the Parkinson's, there is a small section of oral health, but we also mentioned it might be worthwhile to expand this and really look at oral health care in these patients. And I think that is a good thing, right?
yeah, yeah,
Because that drives the field forward and say, look, we don't know exactly what is happening. We, like you say, like other people, you can claim, we can go back and claim, yeah, just take off care of your oral health and you won't develop Parkinson's, which is
yeah.
really not the case. But I think it's good to advocate for these things, right? And say, look, these potentially plays an important role. So when you look at these patients, let's maybe look at oral health care as well,
Mm.
see if that we can maybe run a trial or whatever and see if these things might make an impact. But to come back
Sorry,
to your,
can I just interject on that before
yeah, yeah,
you?
of course, yeah,
Because you know, we know Professor Luigi Nabali.
yeah, yeah,
And I spoke to him on. He's what inspired me to come to you to talk about oral health and inflammation and brushing your teeth. Like these are obviously super important. And should be factored in. I guess what I'm trying to get across is it's not just one sort of treatment or one sort of solution to this. It's such a complex problem where your oral health would then have an effect on your inflammation, and your inflammatory markers may have an effect on your oral microbiome or vice versa. And so it's really understanding all of these sort of factors together and building a very good mechanistic of understanding of how all of this contributes to one disease for now, and then expand it to other diseases. Because at the same time, I'm very big on sort of changing your lifestyle to, let's say, improve or decrease the odds of increasing these sort of biological risk factors for neurodegenerative diseases.
yeah, yeah.
So yeah, as much as I like to make fun of all of these consumer based products,
Yeah, yeah, but there's a difference between very much larger consumer based driven products and
evidence
company
based
and
science.
people, yeah, and companies and people that try to fight the good fight to really try and get somewhere. I do believe that there's a difference between that, but I don't think that is very much different uh, um, in the microbiome field compared to other fields. I mean, people have done this over the years with anything. So there are always people that try to exploit that one way or the other. If it was not just in the scientific world, but in other places as well, right? We see it now with the AI boom as well. I mean, there are some people that try to take the most out of it and just sound nonsense, and other people that try to do the right thing. So it's the same thing. I do think but um, I do going back to the real evidence based things for the microbiome as well, I think is really important. And in this case as well, like I said, the next step for us is trying to get to that point, right? To say we have a very solid mechanism that you can recapitulate even in different models and in different populations, right? So like I mentioned in the beginning, this was a couple of 100 patients, more or less. It wasn't a big cohort, and was from collaborators that we closely have in Turkey. So it's a Turkish population as well. So this needs to be expanded to other populations as well, see if the same holds true. So there's a lot of work, and that's exactly what you refer to, is like really understanding the whole thing, see if this holds true in different models, but also different populations. And then you can get to a place where you say, okay, now we might be able to talk if we consistently see that mechanism, and the mechanism, not just the overall broad findings, which I think is important. Yeah. And
Mm.
like you say, I think especially when it comes to the microbiome, it's multidisciplinary in that sense. It's not at all just either the data driven or just a clinical setting. It's very multidisciplinary in that sense. Yeah.
I could talk to you for a long time, but I feel like it's been too long already. Frederick, it's been a
No, it
pleasure.
was
This has been
great.
so fun.
It wasn't too long as well. It was great.
Yeah, but hopefully we can have you on again soon. I think it'll be really cool. We can talk about different stuff. Yeah, no, this has been awesome. This has
Yeah,
been awesome.
we'll probably have a couple more papers in the next couple of weeks, months. Then we can definitely discuss potentially something else as well. So,
More reading for me. That's what I like.
but it was really great chatting to you. It's great to hear from your side as well. Keep, keep
No,
on keep on fighting the good fight.
yeah. No, thank you.